There are two peptides that come up in virtually every conversation about injury recovery, tissue repair, and healing: BPC-157 and TB-500. They are almost always discussed together, almost always stacked together, and almost always misunderstood because the mechanisms are genuinely different, the evidence bases are genuinely different, and the regulatory histories are genuinely different. One comes from your stomach, one comes from your thymus. They target completely different cellular processes, yet they produce overlapping and potentially synergistic effects on tissue repair. The biohacking community calls the combination the Wolverine Stack, named for the Marvel character whose defining trait is near instant regenerative healing. That name captures the appeal. It does not capture the biology, or the evidence, or the honest limitations. This episode covers both peptides in full. BPC-157 first, origin, all four mechanisms, the animal data, the human data, the regulatory picture. Then TB-500, origin, its unique actin mechanism, the parent compound's human trials, and how it differs from BPC-157. Then the stack, why they are combined, what the evidence actually says about synergy, and the honest picture on what two peptides with no completed human efficacy trials actually represent. Let's start. BPC-157 stands for Body Protection Compound 157. It is a synthetic pentadecapeptide, a chain of 15 amino acids. The sequence is glyglu pro 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 gly lys pro alla asp asp alla glylu val. It was first isolated and described by a research group in Croatia led by Predrag Sikirić at the University of Zagreb in the 1990s. The group was studying a protein found in human gastric juice, the digestive fluid your stomach produces, and identified a specific 15 amino acid fragment that appeared to have protective effects on the stomach lining. That fragment became BPC-157. The name is slightly misleading. Body protection compound sounds like a broad claim. In practice, it refers to the protective function of the original gastric protein. The compound is a fragment of something your stomach already makes, designed to protect the gastric epithelium from acid, enzymes, and mechanical damage. What makes BPC-157 unusual is its stability. Most peptides are fragile. They are broken down quickly by the acids and enzymes in your stomach. BPC-157 is remarkably resistant to this degradation. This is not an accident. It evolved or was selected for in the most chemically hostile environment in your body, your stomach. This stability is partly why it can be taken orally, an unusual property for a peptide, though the bioavailability of oral versus injected BPC-157 remains debated in the literature. The peptide has a molecular weight of approximately 1,419 daltons. It is water-soluble, stable at room temperature for short periods, and requires cold storage once reconstituted. Its effects, as documented across hundreds of animal studies, go far beyond the stomach. The peptide influences healing in tendons, ligaments, muscles, nerves, bone, gut lining, and the brain. BPC-157 has 4 primary mechanisms. Each deserves a separate treatment. The first and most important is angiogenesis, the formation of new blood vessels. When tissue is damaged, one of the primary limiting factors for healing is blood supply. Damaged tissue needs oxygen, nutrients, and immune cells delivered by blood vessels, but the blood vessels themselves are often damaged in the injury. Healing is limited by how quickly new vasculature can form. BPC-157 dramatically accelerates this process. Its primary angiogenic mechanism runs through VEGF, vascular endothelial growth factor. VEGF is the master signaling molecule for new blood vessel formation. BPC-157 upregulates VEGF expression and activates the VEGFR2 receptor, the primary receptor through which VEGF drives angiogenesis. The downstream signaling cascade runs through Akt and eNOS, endothelial nitric oxide synthase. This pathway increases nitric oxide production in endothelial cells, which drives vasodilation and endothelial proliferation, the cellular process of building new vessel walls. In animal models of tendon injury, muscle injury, and wound healing, BPC-157 consistently produces dramatic angiogenic responses. New capillary networks form faster. Blood supply to damaged areas is restored more rapidly. The tissue has access to the raw materials for repair. There's an important caveat here that deserves honest treatment. Angiogenesis is a double-edged mechanism. New blood vessel formation is essential for wound healing. It is also a hallmark of tumor growth. Solid tumors require angiogenesis to grow beyond a few millimeters in size. They recruit blood vessels to sustain themselves. This does not mean BPC-157 causes cancer. The animal safety data across hundreds of studies shows no oncogenic signal, but it is a legitimate mechanistic concern that deserves acknowledgement, and it is one reason the FDA has cited insufficient safety data in its regulatory decisions about BPC-157. The second mechanism is less discussed, but potentially as important as angiogenesis in the context of musculoskeletal repair. BPC-157 upregulates growth hormone receptors in tendon and muscle tissue. Growth hormone itself is a well-established anabolic hormone. It stimulates protein synthesis, promotes muscle growth, and is critical for connective tissue repair. But growth hormone can only do its work if its receptors are present and functional on target cells. In injured tissue, GH receptor expression is often downregulated. The injury itself impairs the cell's ability to respond to repair signals. BPC-157 reverses this. It increases GH receptor expression on tendon fibroblasts and muscle cells, restoring the tissue's sensitivity to circulating growth hormone. This mechanism is particularly relevant for tendon healing. Tendons are notoriously slow to heal because of their poor blood supply and low cell density. The combination of BPC-157's angiogenic effect, bringing more blood to the tendon, with its GH receptor upregulation, making tendon fibroblasts more responsive to repair signals, creates a dual approach to the specific problem of tendon healing. The Sikirić group demonstrated this mechanism in multiple animal models of tendon transection, cutting the tendon completely and measuring healing rates. BPC-157 treated tendons showed significantly faster functional recovery, higher collagen density, and better tensile strength at follow-up compared to untreated controls. The third mechanism runs through nitric oxide, NO. Nitric oxide is a signaling molecule with a remarkably broad role in vascular biology. It is produced by endothelial cells and causes smooth muscle in blood vessel walls to relax, leading to vasodilation, the widening of blood vessels. It regulates blood pressure, blood flow, and inflammatory responses. BPC-157 activates the nitric oxide synthase pathway independently of its VEGF-mediated angiogenesis. Specifically, it activates the SRSE caveolin 1 eNOS signaling cascade, This pathway drives nitric oxide production in the endothelium and contributes to the vasodilatory effects that improve blood flow to injured tissue. The mechanism has particular relevance for gut pathology. In models of inflammatory bowel disease, NSAID-induced gut damage, and alcohol-induced gut injury, BPC-157 consistently protects and restores the gut lining through its nitric oxide-mediated effects. The peptide appears to restore normal blood flow to the gut epithelium, reduce inflammatory signaling, and accelerate mucosal repair. This gastric protective effect is, in some sense, the original reason BPC-157 exists as a research compound. The Sikirić group was studying gastrointestinal protection. The systemic healing effects came later as the researchers discovered that the peptide's mechanisms operate across tissue types. The fourth mechanism is the most speculative but has generated genuine research interest: modulation of the gut-brain axis. The gut and brain are connected through a bidirectional communication network, the vagus nerve, enteric nervous system, and a range of signaling molecules, including serotonin, dopamine precursors, and gut hormones. Disruption of the gut lining has downstream effects on neurotransmitter production and brain function. This is the biological basis of the gut-brain connection. BPC-157 has demonstrated effects on the dopaminergic and serotonergic systems in animal models. Specifically, it has shown neuroprotective effects in models of dopamine depletion, relevant to Parkinson's adjacent research, and has modulated serotonin receptor expression. In traumatic brain injury models and spinal cord injury models, BPC-157 has shown protective effects on neural tissue, potentially through its angiogenic and anti-inflammatory mechanisms operating in the central nervous system. These neural effects are the least well characterized of BPC-157's mechanisms. They represent early stage research with a plausible biological rationale rather than a well-established pathway. But they explain why BPC-157 is discussed not just for physical injury repair, but for gut health, mood, and neurological protection. The animal data on BPC-157 is extensive. It is the most robustly studied peptide in terms of animal model breadth in this entire space. The Sikirić group in Zagreb has published well over 100 studies on BPC-157 in rodent models across 3 decades. The consistency is unusual. The effects replicate across labs and models. Key findings: in tendon transection models, BPC-157 treated animals showed significantly faster functional recovery and better collagen organization at healing sites compared to controls. In muscle crush injury models, recovery was accelerated and inflammatory infiltration was reduced. In gut injury models, NSAID damage, alcohol damage, ischemia, reperfusion injury, BPC-157 consistently protected the mucosal lining and accelerated repair. These effects are perhaps the most reproducible findings in the entire literature. In nerve injury models, including sciatic nerve crush and spinal cord injury, treated animals showed improved functional recovery compared to controls. The 2017 systematic review by Gwyer, Rag, and Wilson in a peer-reviewed journal synthesized the musculoskeletal animal data and concluded that the evidence for accelerated soft tissue healing in animal models was consistent and mechanistically coherent. The July 2025 systematic review published in Orthopedic Sports Medicine co-authored by Vasireddi and colleagues, specifically examined BPC-157 in the context of sports medicine and concluded that the preclinical evidence across tendon, ligament, muscle, and bone models was compelling, while noting that the absence of human RCT data prevents clinical recommendations. Two caveats apply to all of this. First, the rodent to human translation problem is not solved by the volume of studies. More rodent studies do not make the human effect more certain. Second, the Sikirić group dominates this literature. Independent replication is present, but less extensive. This is a genuine limitation. The human data on BPC-157 is limited. The honest picture. As of mid-2026, fewer than 30 human subjects have been studied in published trials of BPC-157. There are no randomized controlled trials, no placebo-controlled studies, no phase 2 or phase 3 trials. What exists? 3 small published studies, all pilot level. The 2024 interstitial cystitis study, Lee and colleagues, treated 12 patients with bladder injections of BPC-157. The results were striking: 80 to 100% symptom resolution in this notoriously difficult to treat condition. This is a tiny sample, uncontrolled, but the result is notable. The 2021 knee pain study, 16 patients, 87.5%, reported significant relief at 6 to 12 months. Again, small, uncontrolled. The 2025 intravenous safety study, 2 adults tolerated doses up to 20 milligrams IV without adverse events, a safety signal, not an efficacy signal. The phase 1 trial registered in 2015 with 42 volunteers, NCT number available, never published results. The reasons are unclear. The FDA designated BPC-157 as a Category 2 bulk drug substance in 2023. This means it cannot be legally compounded by 503A or 503B pharmacies in the United States. The FDA cited concerns about immunogenicity, manufacturing impurities, and insufficient safety data. In early 2026, there were reports of regulatory reconsideration influenced by shifts in the political environment around supplements and alternative medicine. The picture is in flux. The underlying evidence base has not changed. The gap between the animal evidence and the human evidence is wider for BPC-157 than for almost any other peptide in this space. Hundreds of animal studies, fewer than 30 human subjects, no RCT. This gap exists primarily because the peptide cannot be patented. It is a fragment of a naturally occurring protein, which means no pharmaceutical company has a financial incentive to fund the trials needed for approval. TB-500 is a synthetic analog of thymosin beta-4, T-beta-4, a naturally occurring 43 amino acid peptide found in virtually every human cell. TB-500 is not identical to thymosin beta-4. It corresponds to the first 17 amino acids of T-beta-4. Specifically the actin binding domain, the region most associated with the peptide's biological activity. Thymosin beta-4 was first isolated from the thymus gland, hence thymosin, but it is expressed far beyond the thymus. It is found in high concentrations in platelets, macrophages, and wound healing tissues. It is encoded by the TMSB4X gene. It is one of the most abundant peptides in the human body. Its concentration in platelets is particularly high, relevant to wound healing, as platelets are among the first responders to tissue damage. TB-500 was developed as a research tool to study T-beta-4's actin binding functions without the cost and complexity of synthesizing the full 43 amino acid molecule. So the first 17 amino acids retain approximately 60% of the full molecule's biological activity. The complete sequence is required for maximum efficacy, but the truncated version provides a useful research approximation. TB-500 gained attention in the equine performance world before the human biohacking community discovered it. Horse trainers were using it for injury recovery in racehorses, a use that led to its detection in doping tests and contributed to its eventual addition to the WADA prohibited list. TB-500's primary mechanism is fundamentally different from BPC-157's, where BPC-157 primarily drives angiogenesis and receptor upregulation. TB-500 operates at the cytoskeletal level. It regulates actin dynamics inside cells. To understand why this matters for tissue repair, you need to understand what actin is and what it does. Actin is one of the most abundant proteins in the human body. It exists in two forms: G-actin, globular, monomeric, free-floating, and F-actin, filamentous, polymerized, forming the structural cables that give cells their shape and enable them to move. The transition from G-actin to F-actin, polymerization, is how cells move. When a cell needs to migrate to a wound site or extend a membrane to engulf a pathogen or divide, it does so by reorganizing its actin cytoskeleton. G-actin polymerizes into F-actin cables that push and pull the cell into new shapes. TB-500's core mechanism is actin sequestration. It binds to G-actin with high affinity, binding constant approximately 0.5 micromolar, and prevents premature polymerization. This maintains a pool of free G-actin available for rapid deployment when cells need to reorganize. The practical effect: cells can move faster and more effectively. Fibroblasts, the cells that lay down collagen in healing tissue, migrate to wound sites more rapidly. Endothelial cells, which form blood vessel walls, migrate and proliferate more effectively. Immune cells, macrophages, which clear debris and coordinate healing, move more efficiently. This is why TB-500 is described as promoting cell migration and wound healing. It is not directly building new tissue. It is enabling the cells responsible for repair to get to where they need to be and to do their work once they arrive. A 2024 study raised an important question: whether TB-500 itself is the active compound or whether its healing activity comes from a smaller breakdown product produced when it is metabolized in the body. This has not been resolved and represents a genuine uncertainty about the mechanism in vivo. Beyond actin sequestration, TB-500 has several secondary mechanisms that contribute to its tissue repair effects. Angiogenesis: TB-500 promotes endothelial cell migration and tube formation, the process by which endothelial cells organize into new blood vessels. This overlaps with BPC-157's angiogenic mechanism, though through different upstream pathways. TB-500 drives angiogenesis through direct effects on endothelial cell motility rather than primarily through VEGF signaling. Anti-inflammation: TB-500 reduces the production of pro-inflammatory cytokines in wound environments. This creates a more permissive environment for repair, reducing the inflammatory phase of healing and accelerating transition to the proliferative phase where tissue is actually rebuilt. Stem cell mobilization: some studies suggest that T-beta-4 and TB-500 promote the mobilization of stem cells from bone marrow and their migration to sites of injury. If this translates from animal models to humans, it represents a regenerative mechanism beyond simple wound healing, the potential to replace tissue rather than just repair it. The parent compound T-beta-4 has reached phase 3 human trials in ophthalmology, specifically for corneal surface repair and dry eye disease. These trials have demonstrated measurable improvements in epithelial recovery. They do not establish systemic efficacy or safety, but they give T-beta-4 a richer human data context than BPC-157's parent protein has ever achieved. The rationale for combining BPC-157 and TB-500 is mechanistically coherent, even if the clinical evidence for the combination specifically is absent. The logic runs like this. BPC-157's primary strength is angiogenesis, building the blood vessel infrastructure that healing tissue needs. Its secondary strength is GH receptor, upregulation, making the tissue more responsive to repair signals. TB-500's primary strength is cell migration, enabling the fibroblasts, endothelial cells, and immune cells to reach the injury site and organize effectively. Its secondary strength is creating the cellular conditions for rapid cytoskeletal reorganization. At a wound site, both processes need to happen. New blood vessels need to grow. BPC-157's mechanism. The cells that build those blood vessels need to migrate to the site. TB-500's mechanism. The fibroblasts that lay down collagen need to arrive and organize. TB-500. The tissue needs to remain responsive to growth signals while rebuilding. BPC-157. The two peptides target adjacent steps in the repair cascade. They do not duplicate each other. They address complementary bottlenecks. Whether this synergy translates into measurably superior outcomes compared to either peptide alone in humans is not established. The combination has not been studied in a controlled human trial. The rationale is coherent. The evidence for synergy specifically is extrapolated from animal data and mechanistic reasoning. Community protocols typically use 250 to 500 micrograms of each 2 to 3 times weekly for 4 to 8 weeks. None of these doses or durations are evidence-based in humans. The population using BPC-157 and TB-500 is different from the MOTSC or GLP-1 population. These are not primarily longevity researchers or metabolic health optimizers. They are people with specific injuries, torn tendons, chronic joint pain, post-surgical recovery, needs, gut issues that have not responded to conventional treatment. The typical profile, competitive athletes with injuries that are healing slowly. Weekend warriors who have been told to rest for 8 weeks and are looking for something to accelerate that timeline. People with chronic gut conditions, IBD, leaky gut, SIBO, who have found limited relief from standard approaches, post-surgical patients trying to accelerate recovery. The appeal is specific and practical. These are not people trying to live to 150. They are people trying to get back to the gym in 4 weeks instead of 12. The safety profile from animal studies is the best of any peptide in this space. BPC-157 has no established LD50, the dose required to kill half a test population. In thousands of animal studies, no serious toxicity signal has emerged. TB-500 has a similarly clean animal safety record. Both are WADA prohibited. Both are FDA Category 2, cannot be legally compounded in the US. Both are available as research peptides from overseas suppliers operating in a gray market. Here is the honest picture on BPC-157 and TB-500. BPC-157, the most extensively studied healing peptide in animal models. Hundreds of studies, consistent results, mechanistically coherent, and fewer than 30 human subjects studied in published trials. No RCT FDA Category 2, WADA banned. The gap between preclinical promise and clinical evidence is enormous. It exists because the peptide cannot be patented. TB-500, a synthetic analog of one of the most abundant peptides in the human body. Its parent compound has reached phase 3 trials in ophthalmology. The actin sequestration mechanism is well characterized. No human efficacy trials for systemic use. WADA banned, FDA Category 2. The human evidence base is thin, even by the standards of this space. The stack together, mechanistically coherent, potentially synergistic. No controlled human data on the combination specifically. The question everyone in this space is asking is, when do the human trials happen? For BPC-157, the answer is probably not soon without a major shift in research funding. The patent problem is not solved. Academic centers would need to fund the trials independently. That is slow, expensive, and rare. For T-beta-4, the parent compound, the ophthalmology trials are an existence proof that human trials are possible. Whether systemic trials follow depends on whether the pharmaceutical companies or academic centers see a viable path. For the people using these peptides right now with specific injuries based on compelling animal data and community experience, the honest assessment is that you are operating ahead of the evidence. The mechanism is plausible, the animal safety profile is clean, the human evidence is absent. That is the complete picture. Subscribe to Peptide Decoded, new episode every Tuesday. Drop a comment. If you have used BPC-157 or TB-500, what were you treating and what did you experience? We read every one.